Greetings, germophiles, and welcome to a live edition of MicroTalk. So we have normally a podcast that you guys, if you're true germophiles, you're going to listen to this, right? But we're doing a live version here at ASM Microbe, and I'm Karl Close. I'm at the University of Texas in San Antonio, and I usually come with my colleague, Dr. Janikiram Seshu. But sometimes Dr. Seshu has someone taking his place. Who's here today?
Hey, what's up, Germophiles? This is Seshu, the awesome undergraduate student majoring in microbiology and immunology at UTSA. I'm kind of representing all the undergraduates of the world who are interested in microbial sciences.
And in case you somehow missed that, he's also a puppet. So he comes with that perspective as well. So it's a very unique perspective. Anyways, we are, of course, excited because we have two of the, pillars of microbiology here sitting between us. And that is two friends of mine, actually. And one of them is Dr. Steve Finkel. He is at University of Southern California, right around the corner from here, actually. And also Vaughn Cooper, and he's coming to us from University of Pittsburgh. Great to have you guys here. We have past and future ASM presidents. We also have world experts on microbial evolution and adaptation. And we have world experts on Frank Lloyd Wright and on triathlons. So we have lots of stuff to talk about. Great to see you guys, obviously, right? So Steve can tell us even a little bit about Los Angeles. If you're not from Los Angeles, he can tell you a little bit about this town. He's lived here for a long time. And anyways, great to see you guys. So Steve, tell us from your perspective, what do you see sort of like the evolution, since you're into evolution, of ASM from when you started to currently, and what Vaughn is going to be inheriting?
So I actually checked my notes. This is my 20th ASM national meeting.
Amazing.
When it was the general meeting, and now it's Microbe. Lots of things have changed. I think in terms of the really good change, I think the society has taken on the responsibility, as they should, to really try to bring our younger scientists in much more than we used to. In my day, I think graduate students were there to be maybe seen but not heard and to do their posters. And one of the things that I really have been enjoying, especially since COVID and we came back, was how actively we're asking graduate students and postdocs to participate, give talks, pull people from posters to give talks, and then all the other activities here with the track hubs, which I absolutely adore, and how we've really tried to bring everybody in and get the more senior folk, more junior folk together in maybe slightly more casual ways to interact, but still with a little structure.
Yeah.
And I think it's been very successful. And
awesome.
It's certainly something I've been enjoying.
And so did that start when you were president, or is that something that happened?
People have been talking about it for a long time, but I think during my presidency, it was the first back in person microbe meeting. We had been off for a couple of years because of COVID.
Right.
So Washington, D.C., my president year was the first time we were all back together. And it's the first time we really start to see that. I would go to regular sessions, and there's always grad student or postdoc speakers along with the more established folks, and I thought that was fantastic.
That's awesome. And Vaughn, you're going to be inheriting this, evolving, event, let's say, and the evolving society. How do you see like the progression of ASM, and do you have like ideas about where you think it's going?
So I think that the big change that's coming, right, are the three new scientific units that we've established have established and are establishing at ASM, representing health sciences, fundamental sciences, and applied and environmental sciences. So that's effectively the model will be 3 concurrent meetings with opportunities for crossover. And I think that crossover remains really critical because we both you use the word evolution more than once. Yeah. I was trained as an evolutionary biologist even before a microbiologist. Steve is really one of the other evolutionary microbiologists who's held leadership. folks like us in this role in the society are relatively new, just as this new structure that we're proposing is relatively new. What we hope that does is actually provides even more of a home, more opportunities for the trainees to be involved, postdocs to get involved, young faculty to be involved as presenters, as planners, right? As meeting planners, and get involved in those areas of science and ultimately support the functioning of those meetings so that they interact.
Okay.
So, Dr. Cooper, I'm going to start with you. If you're going to be putting out a TikTok video for undergraduates to get excited about microbial sciences, what will be that one topic that you will go after? And maybe Dr. Finkel can answer the question after that. It has to be only one, and it's a TikTok video. It can't be too long.
The origins of life.
Origins of
life. Yeah. So we just had this really terrific symposium last hour, sponsored by the Moore Foundation. We held 3 colloquia. We had a synthesis meeting about that. What my co-chair Mike Lynch said is it's one of these big questions that we spend a lot of time thinking about the origin of the universe, and we spend comparatively less time investing in this deep question about where our life came from. I need to tap the undergrads in my lab, maybe along with you, to make that TikTok, because I'm older than I look. But
okay. We can help you.
Whereas I am as old as I look.
I'm going to say maybe
older.
Yeah, a little bit older.
Thank you, Dr. Close. Vaughn took one of my answers. The thing that I've told people over my entire career is, so I tell your TikTokers, is microbiologists think they can do anything. And the good news is we mostly can, and we're relatively inexpensive. And when we mess up, it's usually okay.
Yeah.
So, if there can be a TikTok around, really cool science that if you mess up, it's okay.
Okay. That's really, really, really important for undergraduates because we always mess up.
Especially puppets. They don't, because they don't really have good manual dexterity. It's a problem in the puppet community. So I want to talk to Steve and Vaughn about the challenges. I mean, we see positive things, right? But there's a lot of challenges right now to just science in general, and I'm going to say also to academic scientists as well as industrial scientists too. How do you think that the ASM can help or promote microbiology in the face of the various things that we're facing?
So I mean, I think it starts from the place that brings us all here in the first place, which is that we love our science, right? We, in spite of scarce resources, just like Margie Lee said at the opening address last night, it's possible to do a really great microbiology, and that's always been true and always will be true. I think this still is the best ever time for microbiology, and I said that 2 years ago, and I said it 5 years ago. It just keeps getting better to be a microbiologist. That's true whether you are flush with resources or not.
Right.
I think that will prevail. That also motivates us to get together, band together to campaign for obviously a better research environment.
Absolutely. Steve?
One of the things that buoys me and has since I became, a practicing microbiologist really as an undergraduate is this community. And there've always been ebbs and flows, and I think, right now may seem a little more intense and a little more negative than maybe it's been in recent memory, but the community is tight. And we have a culture of helping each other that I've experienced from undergraduate to today. And it continues. And, when I've had some issues or other colleagues have had issues, people have really been incredibly generous with their time, with their ideas, hopefully with some money. And, and there's so many people we can rely on, and strangers too. Someone will say, talk to Steve. And, so I'm getting people who I don't know are reaching out, but, I want to give back. I think those of us who've reached this position, we have a lot of responsibility to help,
Sure.
more junior folks, but all of our colleagues. And that's how we get through it. We're in this together. The work keeps going forward. We do the best we can with what we have. I think we have to advocate for our work in ways we didn't before, and talk to whoever will listen. I think if you heard the panel just before, I think maybe it was Alex or Stefano was talking about just talking to whoever will listen. And we have to do that more and advocate for ourselves more than perhaps we have in the past. But I think with the strength of the ASM, with the strength of our advocacy, and then just the microbiology community in general, we have a long and I think proud tradition of helping each other out.
Yeah, yeah. Well, and I think one of the things that's obvious to me is that, both of you are really dedicated microbiologists, and you feel sort of a responsibility maybe to the microbiology community. I mean, you can see that, right? And the other, like Terry in the audience and Stanley over there, like the people who are officers in the ASM are really dedicated people that really have a strong desire to, help the community. But my question for you is, how do you get the next generation to sort of fulfill those roles?
So, I think the best way to do it is to let them be microbiologists themselves as young as possible. Okay. As you know, that's been a mission of our program that we call Evolving STEM, to reach kids in middle school or high school and let them actually do research. That's a great start. And it doesn't have to be our program. There's lots of other simple and safe programs that allow kids to actually see themselves as potential scientists, hopefully microbiologists. And I think microbiology is like the ultimate gateway, science, right? As I keep saying, it's pretty simple, especially if you put, for instance, triclosan in the media so that nothing but Pseudomonas can grow. It's a nice trick. But I want to say, just in addition to that, I'm always amazed by how many folks here at ASM are innovating their education because of their research, and then that feeds back to their research. There's a tradition of that here in this community. We are good at building our own, and we try to do it as early as possible.
That's awesome.
So,
do you have any ideas on that too?
No, it's just, as a follow-up of that question, one of the psychological barriers undergraduates have is that we don't want to mess it up. We don't want to do something that we do not know is going to work out. So what was the dumbest thing that you did as an undergraduate that is scientific or microbiological? And give us an example so that we can also continue doing it.
I have a good story. Yeah, thank you. I'm taking this one. So I was working on a virus as an undergraduate that killed gypsy moth caterpillars. And we were trying to basically evolve the virus, which means we have to raise the little, neonate, new gypsy moths from larvae, from eggs. And we keep them in an incubator, and you have to keep the incubator nice and tight, because if they hatch and they see light, they are phototactic and they will climb out. And the gypsy moths make these big silken webs everywhere and make a terrible mess. They became a problem because they were imported to make silk. So one time I was in late doing an experiment. I didn't quite latch that incubator tight and set up a hatch. And I got a call right when I was earlier than I wanted to be awake, and at like 7 a.m. from the lab manager hollering at me because there was only one person who could have made that mistake, telling me to come in and clean up. And so I had to skip the morning to clean the lab, and I was covered in gypsy moth larvae and silk for longer than I care to remember. But I still got my thesis, so
okay, yeah, that's good.
I didn't get kicked out.
You were certified dumb?
Yes.
Maybe.
My undergraduate project was I was an undergraduate at Berkeley working with Rich Callender in a phage lab, and my job was to complete the DNA sequence of the non-essential region of bacteriophage P4, which is the best of all the phages. It's a satellite of bacteriophage P2, for those of you aficionados who might not remember. So the genome had been sequenced, except for the non-essential region. So we'll let the undergrad do that. It's 2,500 base pairs. It took me 2.5 years. because I had to subclone everything down to something that you could sequence across, and we only had one primer in those days. But I had MP18 and MP19, so I could clone it this way, and then use one primer to go across and get double-stranded. So as I was getting better and better at sequencing, I needed longer and longer gels, and everyone else in the lab just used the standard gels. And the undergrads and grad students here will not know what I'm talking about. But I ended up, I needed to run longer. It was working great.
Yes.
Well, that's true too. Anyway, I needed to run longer and longer gels, and we had these 1 meter plates. So I found a gel box that I could do 1 meter plates, and I set it up. No one in the lab knew how to do any of this, and everyone was using all the power supplies. So I needed a power supply. And there was this one power supply from the 50s that was off in the corner. And it only had one dial. Usually it had, you could set current, you could set voltage, you could set wattage. This thing had no meters and just one dial. It was called the Killdozer, I remember. And I set it up, and I know what wattage it's supposed to run at, and no one could really guide me. And I remember we'd started it just before dinner. So I set it to what we thought was the right current, and I saw bubbles in both chambers, so I knew that it was working, and we went to dinner. And we came back an hour and a half later, and the lab is full of smoke. And I smelled something that no biologist is ever supposed to know the smell of. I know what burning acrylamide smells like. Yeah. So I did not start a fire. I just started a smolder. But that was probably my dumbest. I set it to 11.
I was going to say they were to 11.
I set it to 11, yes.
And you still got your degree. So
I still got my
there you go.
degree, and
Yes.
I have 2 children who have 10 fingers and 10 toes, so whatever
Right.
exposure
Right.
happened. By the way, in those days, we used to load our radioactive gels by mouth.
That is absolutely true. Yeah. Yeah, Sydney taught
That's
me
how I was
do
taught
that.
to do it.
Yeah.
Sydney taught you to do it. Rich
did.
taught me to do it.
Yeah.
Yeah. Remember the thing with the red side and the white side? You never put the red side in your mouth. You only put the white side in your mouth. I'll explain it all to you later.
So we're these guys here are also world experts in evolution, and so I want to know where is the field of evolution and adaptation. So let me take a step back. So Steve studies evolution under stationary phase conditions, and many of you, I don't know, maybe you don't know that there are these cultures of E. coli that have been sitting in a tube for long periods of time. And I think I'm going to say the prevailing thought before was that they're just sitting there, nothing interesting is happening. And Steve has proven that that's really not the case, that there's evolution going on under those conditions. And he's discovered this interesting phenomenon called GASP that has to do with basically competition under those stationary conditions. So how does the study of evolution under those conditions influence our understanding of evolution in general?
And I didn't discover GASP. That was Roberto Coulter coined that term. So I just want to give
he's
my
not here. You
I want to
can
give my
just say
postdoc mentor the credit.
He's not here. You could just say
I just did it longer than anybody else. Yeah, I mean, the fundamental observation is that the bacteria that come out of the test tube after a fairly short period of time are different than the ones that we put in, and that we could keep them going for ages without adding any food, and that there was really great adaptive change. So it's become a model for other systems and for natural systems. I mean, there's lots of ways to apply that kind of thinking, and Vaughn knows way better than I do because I'm actually not trained as an evolutionary biologist. I'm a total accidental evolutionary biologist. I just had data and bacteria doing things. He actually understands the theory. But I want to tell you just to illustrate how our thinking has really helped us understand in a natural environment. Mm-hmm. I'm really very fortunate to be able to work with geomicrobiologists and marine geomicrobiologists who get water samples from really interesting places, like half a kilometer beneath the floor of the Atlantic Ocean. Right, and there are these, essentially wells down there, and then you go down there with a remote operated vehicle or with the Alvin submarine, and every so often you're allowed to pull up these bottles and get this water. And we just actually published a paper recently where we got bacteria that came out of those waters. They're called crustal fluids, and there were some halomonas that grew that came out of these waters, and there was not very much dissolved carbon, but the microbes were there in reasonable numbers, and they would actually make colonies on plates of agar with no added carbon. So they're incredibly exquisite scavengers. And we thought that was really cool, and we wanted to see if we could figure out how they did that.
Yeah.
So we took our, more dumb E. coli-based experimental approaches and did what we always do. We see if something will grow in LB. And usually microbes that come from really low nutrient environments don't like rich medium. These halomonas said fine and grew to the same density as coli. So we had a crazy idea, but fortunately it worked. We said, let's adapt these microbes to growing under high nutrient conditions and then see if they, quote unquote, forgot how to grow under the really low nutrient scavenging conditions. So
Classic experiment. I like
former graduate student of mine, Hans Sebastian, did this experiment, and he set up a bunch of parallel cultures and did serial passages under high nutrient, and then he went back and put them in the environment, which is just salt water, basically. And we got mutants who couldn't scavenge anymore. They grew just fine in LB, and they couldn't scavenge anymore. And we did this in a dozen independent experiments, and we kept hitting the same loci with different mutations. So they were each getting hits in the same loci or the same pathways, which is now hopefully helping us understand what activities are needed to live in waters that are between 2,500 and 250,000 years old. The geologists can argue about that, and the geochemists can argue. But identifying genes that might be required to survive under these really, really, truly low energy, low nutrient environments. So we've been able to take what we learned from E. coli in a test tube evolving to a real environment.
Well, I thought you were going to say that, the analogy is to Americans that, just eat junk food or whatever. They don't know how to, maybe just sit on the...
That's a different project with a worm biologist where we give them American diet. We take that's nutrient NGM nematode growth medium plus sugar. That's we call it the American diet.
Okay.
So, as evolutionary biologists, were there any mutations in your career plans that changed the course of your own personal evolution, for good or bad? What are the 2 points, or maybe these mutations are rare or frequent, that changed your course of your careers?
So,
spontaneous or induced?
Spontaneous,
well,
induced.
so I mean, Steve gave a really lovely sort of synopsis of the playbook where you design these evolution experiments, you see what happens, you measure changes in phenotypes, and then you see whether they have consequences on their environmental effects. We did something similar, really, when I started my lab back in New Hampshire more than 20 years ago, and we were using a strain of Burkholderia.
When he was 12.
Yeah, exactly. When we were using a strain of Burkholderia that came from the soil, and we were asking how does it adapt to maybe form biofilm as a way to model how Burkholderia might adapt when they establish chronic infections in persons with cystic fibrosis. And that was right when, so not only did the experiment work and we saw these colony morphologies that were indicative of producing more biofilm, we picked those clones and we sent them to the Joint Genome Institute, who had just established a community program to help us use Illumina sequencing, which is brand new, to identify genotypes. And we discovered that these colonies had mutations again in the same gene. I'm still studying that gene, and we've learned that gene, when mutated, is associated with worsened lung function in persons with cystic fibrosis. So just really by chance of modeling this biofilm, we happen to find this regulator, and it turns out that regulator is broadly important for how Burkholderia adapts in CF. And so there's sort of cool stories about the genetics there, but it's also about the story, as you were asking, like our fundamental work in the lab about how biofilm evolution happens motivated us to do more and more work with clinical samples where people have chronic infections. You can use the same approach where you just pick samples from sputum or what have you over time, and you ask what mutations are accumulating in those lines. And if they happen in multiple patients, in sort of in parallel, it gives you the same basically readout that you get from these evolution experiments in the lab.
Cool.
So, kind of following up on that, I want you to kind of expound a little bit on the idea that that if you're, say, in the lab, right, and you have experiments running, and something weird happens, like a weird result that you weren't anticipating. The value in having an open mind and following the lead rather than just throwing it away because it didn't meet your preconceived thought. Can you talk a little bit about that?
Yeah, I mean, I think any of us who do evolution experiments are always following, the cool oddball things because, it's acting on natural genetic variation. You might have an idea what's going to happen, but I guarantee you Steve had no idea that he was going to find this regulator from these deep sea microbes, and I didn't know I was going to find this regulator. I didn't know it was going to produce these colonies. In fact, the weird colony morphologies were rare. So if you were really strong-willed, you might even think they were a contaminant or something not to pay attention to.
Right.
But I think one of the benefits of sort of this ecology and evolution background is you have an eye for diversity.
Yeah.
And that's the thing that you wind up following up on.
Right. And so those are kind of inflection points, right? I
mean, they really are. And I'm not sure if I landed it, but like that inflection point is what actually drew me into medicine more so than the basic science but it's the same through line.
Yeah.
But your point's really important about, recognizing the diversity as an attribute instead of as a problem. And because I didn't come out of population biology and population genetics and evolutionary biology, and I was trained to be a hardcore reductionist prokaryotic molecular biologist, when I started aging these cultures far longer than I was quote unquote supposed to, and this was a hobby as a postdoc, and I started seeing colony variants, my first thought was they're contaminants. But then you think about it. What microbe from my mouth, my fingers, the ventilators in our 1912 building would want to live in 60, 90, 150 a year old culture of spent LB and take up residence? Fortunately, I had the insight to start with 2 strains which were each resistant to a different antibiotic. So when I got the quote unquote oddballs, and then these are all resistant to strep, or these are all resistant to now. So they were descended from the parent.
Right.
And then that's when the light bulbs went off. And it was like, oh, this is really cool.
That's
and
awesome.
then you talk to people like Vaughn, it's like, yeah, that's what happens.
So I am 21 years old right now, and
He's
I plan to
aged
work
terribly
on.
I
plan to work on another 50 years on science. So I want the 2 presidents, or former and the current or future president, to tell us how wrong they were today as to what I will be doing 50 years from now.
So I think you'll be able to work with synthetic microbes, right? You'll be able to work with engineered genomes and likely engineered cells, or at least partially engineered cells, to ask any other question you want. And maybe not cells, cultures, communities, diverse communities, to build things, to test fundamental questions about how microbes work, to hopefully develop the next generation of climate capture or climate remediation tools.
Or would I even have a job, or maybe it's already taken over by AI,
the robots?
That I don't have to really do anything.
Do you want to comment on that?
Well, I can't comment on whether or not there are going to be jobs. I can comment on whether the AI bots are going to take over. And I don't know what AI is going to look like in 50 years. But based on what I've seen currently, we all still have job security, right? It's a tool. We've had a lot of tools that were going to revolutionize everything and put certain techniques out of job. Genomics was going to kill genetics. Well, nope. Turns out, no. I would have loved it, but it's not happening. We still need genetics. And so I'll say yes and to what Vaughn said. I think everything Vaughn said is going to happen. But I think fundamentally, there's all these tools, but, PI initiated. I mean, I'm assuming there's still research run universities 50 years from now. Stefano? No comment. That there's just, we don't know where the next good idea is going to come from. And when you think about some of the things people like to point to as amazing tools or amazing advances that have come out of microbiology. So let's just pick two. Genetic engineering, cloning was invented, obviously based on tools isolated from bacteria. And CRISPR's the other one. And CRISPR's really the best example to point at where you don't know where the next great idea is going to come from. Because at the time, the folks who were doing that research had no idea.
Right.
They were doing their thing. And then it was sort of forgotten for a good long time. People continued working on these things.
Yeah.
But it was insights decades later. And if we don't continue to promote that kind of research, I worry about what we're going to miss.
Right. No, that's great points. I want to open it up to questions in just 1 second, but I also want, one of the things that we try to do in microTalk is to humanize scientists, because I think there's some mystique about scientists. And I just want Steve just very briefly to tell you about his passion about Frank Lloyd Wright, which, everybody has a passion, right? And I think it's really amazing that he knows so much about this one particular architect. But can you just give them a flavor for how important, that sort of hobby is to you?
And Karl is overselling my level of expertise.
Not at all.
I mean, compared to Karl, I know a lot about Frank Lloyd Wright. I'll take that. I'll take that.
He does.
I have a very long-standing interest in architecture of all kinds. I've had a very long-standing interest in what we now call modern architecture or mid-century modern architecture. Going back to when I was a kid, I'm originally from New York. I got to go to the Guggenheim when I was a kid, and I thought that was a really cool building. I wasn't so much into the art at the time, but I was really into the building.
Cool building.
And then I had the good fortune of being a graduate student at UCLA, and there are some Frank Lloyd Wright buildings in Los Angeles, which I got to visit when I was in grad school. And I started reading more, and I'd see a cool building around LA, and it was either a Frank Lloyd Wright building or one of his apprentices.
Yeah.
And there's a lot of in Southern California, within a few miles of here is like the center of mid-century modern architecture, which really started in the 1920s in LA. And then the stuff we think about, what the 50s and 60s look like, the Jetsons and the Incredibles houses and all that kind of thing.
I told you he knew a lot.
Anyway. So, it's a nice hobby. And the good thing is whenever you go to a city in the country, there's a chance there's something within 50 miles.
Sure.
I've dragged Karl to places in, small towns in Wisconsin.
Yeah.
And it's and also one of the things that really appeals to me about Wright is he really thought outside the box, literally and figuratively. If you think about open plan architecture, everyone wants a ranch house with open plan. Frank Lloyd Wright was the first person to build domestic homes that didn't have every room had walls. If you like carports, Frank Lloyd Wright invented carports. He didn't like garages because he knew people put junk in them. So you just need something to cover your car so it doesn't get snowed on. So he invented carports. And there's a million things we take for granted now that he innovated or his apprentices did. And so if you're open to new ideas and you like to experiment, he's a fun architect to think about.
Well, and I think that there's a biological connection there, right? Because what I've learned about Frank Lloyd Wright is that the architecture was always meant to be sort of like basically woven into the environment that it was in. And so similar to microbes that, I don't know, I'm making an analogy out of nothing. Vaughn is an athlete. You can just tell by looking at the guy. He's a natural born
and you can
athlete.
tell I'm not.
I didn't say that, but I'm glad you did. So Vaughn is an athlete, and the guy does triathlons. You want to talk a little bit about just that hobby and how that's influenced your, kind of...
I think where I'd start is that it almost got me thrown out of grad school because I was spending a bit too much time on long rides away from the lab at Rich Lenski's lab at Michigan State. But he was incredibly patient, and I had the good fortune of sort of having a story almost ready to share when he was finally had it with it. But I mean, I'll say that the major pieces. It's an awesome community, just like microbiology is. I've seen 2 ASM board directors swimming at our hotel pool here.
Nice.
We could certainly continue our conversations there. Being out on the bike, you can have long conversations about all sorts of stuff. I've had awesome. One of my old riding buddies was a veterinarian who would love to nerd out about the microbiology he would see in his clinic with me on the bike. And the same thing for the runs. And I think running is a great way to explore as you go around and travel as an academic. So, I mean, basically, it's just a way to kind of formalize outdoor recess. And I think when you can make the outdoor recess exploring and social, it's pretty self-sustaining.
That's awesome. All right. So, hey, anybody have any questions for either one of our illustrious guests today, either Vaughn or Steve? There's swag. So if you ask a question, you get something kind of cool. I mean, at least, okay, I think it's cool. Somebody's got a question though.
Karl Close, cool.
Isn't this cool? Come on.
Thank you
Kinda
very
cool.
much for the really great conversation. I was there listening to you and thinking, Wilmore Webley from University of Massachusetts. I was there and just seeing the excitement. It's that excitement that got me into microbiology many, many years ago. And as you think about the future of microbiology and the unanswered questions that are still there within your sphere specifically, what are those and how do you inspire a new generation of microbiologists to continue that trek down what I find to be very inspiring and exhilarating?
So I think that, thanks for the question. A lot of folks come into my lab, which is in a medical school, wanting to solve some question about, the evolution of antimicrobial resistance, which is a daunting challenge that has galvanized a lot of us here. And I think ASM is doing an excellent job sort of nurturing that community and promoting it by promoting interdisciplinary perspectives around it. So the cool part is that gets the students in the door, the trainees in the door. They come for that, but they tend to stay for this discovery of diversity and weird mechanisms that come up from the studies, and that keeps them in the field, even if the funding for that particular, say, drug resistance mechanism passes or by. They've been captured by the passion of the way of using sort of evolution as a tool for discovery. That's never going out of style. Yeah.
Something that it began as anecdote, but over the years, I think it's probably real, and y'all can talk to me. We can argue about the proportion of what I'm about to say. And also, I now have the privilege of serving as our Dean of Graduate Studies in our College of Letters, Arts, and Science. So I get to see it not just in the STEM fields, but across the humanities and the social sciences. I've always put incoming graduate students in, and really the undergrads too, into one of three boxes. But let's talk about the PhD students coming in. About a third of them know exactly what they want to do for graduate school, and they're right. And about a third of them know exactly what they want to do for graduate school, and it turns out they're wrong. They just don't know it yet. And then a third of them really don't know what they want to do for graduate school. They want to earn their doctorate in whatever discipline they've chosen, but they're open-minded. And I know examples of all three. I have one of my grad students is sitting here looking at me right now, and he's category one. He said, "I'm coming to your lab to work on this," and he was right. Okay, great. But so how do you maintain the excitement for all three kinds of folk, right? And it's really by example. I mean, and you just have to, once people believe that you are going to help them get the tools they need to be successful, and that they're going to be in a supportive environment where they can make mistakes and forget things, especially when they're more junior. And if you're fortunate enough to create an environment where you're allowed to mess up and not be reprimanded, and we've done some pretty stupid things, and I didn't talk about the dumb things I did as a graduate student. So I think if you can maintain that, and hopefully you've got more senior folk around who can are walking the walk, and that's good. So I think if you can do that and just keep that in mind. But also, everyone's very different, and we just have to sort of roll with it. And again, because micro is more forgiving, I think, than other disciplines, I don't want to, I'm the only real microbiologist in my department. I'm surrounded by microbiologists at my university. But, in my particular program, we have a lot of folks working on, other systems, and maybe they're not quite as forgiving, or they're more expensive. So maybe I just don't worry as much, because We've got lots of toothpicks that we've been reusing since 2000. As I've been telling people, they're not clean, but they're sterile.
All right, who else has a question? Who else wants some good swag? Don't all jump at once.
Someone in the way back.
Way back.
Way back?
Way back. Zach, you got a question?
Oh, she wanted swag. You have to ask a question for swag. All right, this is a ringer.
Karl, as you mentioned beforehand, being a ASM president is something that takes a lot of responsibility, and it's something that you really want to do. I guess what I'm trying to ask is what inspired you? What made you kind of want to take that next step to reach ASM leadership? Because again, it's a big deal, and it's not easy to do. So what drove you to kind of get to where you are now?
I mean, honestly, I said it earlier, it's the best ever time to be a microbiologist. There's awesome opportunities for discovery and many big challenges ahead for humanity and for the environment, for climate, what have you, that microbes are going to play a part of. And this society is the place to galvanize that. The structure that we're working on building is intended to make that better, easier, more inclusive, bigger tent. It's a challenge that's, I mean, yes, a little daunting, but how can you say no if you sort of believe in the truth of the science?
I did want to point one thing out, though. There was something implicit in your question that I don't fully agree with, because you said strive for. Yeah. And I think based on, I was very proud and honored to run, and I was very delighted when I was elected. That was a huge thrill and one of the highlights of my career. But it's not something I was striving towards. I was serving the organization. I was always participating, but I wasn't seeking the office. And I'm not sure any of us who have been presidents, at least in the last 6 or 7 or 8, were really seeking it. It found us. Now, that's a huge compliment. And we were all doing lots and lots of things and involved with the society for decades, being reviewers and being editors and being on committees and stuff like that. But I don't...
Being complainers about stuff that we want to
fix.
Being
vocal about things and being noticed and being constructive. Yeah. Mm-hmm. So I think it's what's really important, and I think one of the reasons certainly why under current staff leadership and with the current the last cycles of presidents and treasurers and secretaries, these are all folks who really want to give back, and we are where we are today because of ASM. Has been a large part of where we are, and we want to give back. But I don't think any of us were striving towards it. We were just kind of excited when it happened and would have been equally relieved had it not happened.
Yeah, and I guess I'd also say it is, it's a monster honor to be, as Steve said, and then any sort of misgivings or nervousness you have about it is allayed by the fact that this is an awesome team, and all you're doing is just taking a little extra wind in a big team. You got awesome support from really. I mean, many of us are part of other societies. You're not going to find staff that's any better.
And before the next question, I want to say it is about the team. And at any level, whether you're a student or a postdoc or in industry, in academia, there's all this support around you. None of us does this alone. So anyone who's interested in learning more about the organization and giving back, I encourage everybody to ask, because if you ask, people usually say, yeah, come on. You want to help out? Yes, please. And they're going to help you learn how to do it. Hill Day, if you ever want to get involved with Hill Day and you get this great training. So it's a wonderful environment to help us really be as effective as we can be.
Awesome. Do you have a question? Yeah.
2, 4. Hi, afternoon. Paul Brown, University of the West Indies, Jamaica. It's a question of paradox. Now we have two evolutionary microbiologists or biologists. And one of the challenges for ASM is trying to predict or forecast into the future, at least 5 years at a time, what is likely to be the where microbiology should be. But your experience in evolution says that you can't even begin to consider what might be any of the possibilities. So how is it that your experience then with evolution now shapes how it is that you project into the future? But you can't leave everything to chance and what genes are going to be upregulated, downregulated, so to speak, in terms of the activities of the different facets, etc. But you must plan, you must strategize how it is that you think about the future of ASM with respect to evolution.
No, I think that's such a brilliant and fun question. And so let's start with the experimental and use that as an analogy. Yes. We know enough about experimental evolution to know we can't predict exactly what we're going to get. But we try to design our experiments as best as possible so that when we do get phenotypic change, when we do see something that might look like adaptive evolution, we can one, we have to be able to recognize it. And two, we have to be able to characterize it. And three, we can constrain things a little bit. So at least we're not studying everything under the sun. And we can set up, if it's the system that Vaughn cut his teeth on, where it was a really chemically simple system, where it was just really, buffer and glucose, or my system, which is a little bit more complex. But we understand some of the limitations, and that helps, the hanging out with geomicrobiologists, I learned the idea of constraints, and would help us understand what the constraints are. So you don't know what you're going to get, but at least you've designed the experiments to hopefully give you things that you can study and you can observe. And when we're successful, we know because we're figuring little bits out. So I think you can apply that thinking to running a large organization. And, and Stefano talked yesterday about the 2040 exercise, which I was part of in 2023. And with the right kind of folks helping you think about the future. The first question we got asked, or first question I asked, how am I supposed to think about what's going to happen 17 years from now? And we just sort of, initially I was paralyzed. And the folks who were helping to moderate this, who Stefano brought in, said, well, think about where your science was 17 years ago. And what were you wishing you could do in the future? And I could remember where we were 17 years ago, and I could remember what I was asking for, and some of it actually came to pass. And that really liberated me to think about, so there are some things that we can think about that might not be pie in the sky. Some are pie in the sky, some are more rational. And I think what you just have to do is think about what is possible, understand what your limitations are, and then just go from there. So whether it's a flask or a test tube, or whether we're going to have a budget of X million a year available, Y million a year available, or we're going to be a deficit situation. What can we do with what we've got? And then I also think about Dr. Lee yesterday as well. And again, there's a reason we keep 5,000 toothpicks handy at all times.
Well, it's been great. I want to thank Steve Finkel and Vaughn Cooper for having a nice discussion. If you guys want to talk to them, you can come up now. We got some extra swag too, so if you want some of that. And thanks for listening.
Karl is saying we have to pay you to talk to us. I just want to make that clear.
Pretty much. Anyways, and we will hopefully have some of you as listeners on our microTalk podcast. And thanks for being here today.
Thanks very much, Karl.
Thank you.